We've already talked many times about proteins throughout the course of lecture. However, now we're looking at the structures of proteins. We've looked at catalase in laboratory, that is an enzyme, but it's a protein. We've looked at biuret, which is an indicator that tests for the presence or absence of proteins in lab. So you've been introduced to proteins, but as far as proteins go, when we look at biological molecules, these are by far the most diverse of all of those biological molecules. Everything from the action of enzymes clear down to communication with cell signaling. These molecules, these macromolecules, are involved in so many things with the cell. Going back to that whole thing with monomers coming together as units that are similar that make and build polymers. Amino acids are monomers, units that are similar that come together to make polymers that are called polypeptides. So when you look at this slide, starting on the left, the orange box is an amino acid. On the left-hand side, you see the atom, that's the N, bound to two hydrogens. That's the what functional group? The amino group. On the right-hand side of that yellow-orange box, part of that circle, that OH, is the hydroxyl group. But if you look at that whole functional group that's part of that, that's the carboxyl group. The C, that's double bound to an oxygen, that carbon that's also bound to that hydroxyl group. That's the carboxyl group. That is an amino acid. Each amino acid is similar. Okay, they have very similar units that come together, and when those monomers join together, it will make the polymer that is called a polypeptide. The bond that joins each amino acid occurs when water is removed. So when you look at this diagram, you've got the one amino acid on the left-hand side with the hydroxyl group that then binds with the hydrogen of the amino acid that is in the middle. Water comes off, those two bind together. Now where you see that covalent bond between the N and the C, that is a peptide bond. And then the next bond that occurs between that middle amino acid and the third one, where water is removed again, that is another dehydration reaction, and another peptide bond forms. That forms a polypeptide. Now if you notice the letters that are associated with those three amino acids, they have little individual names that we go by. We shorten those names. These three, the one on the left is called alanine. We call it ALA. The one in the middle is glycine, GLN. And the one on the third one in the pink is isoleucine, ILE. Okay, so in this whole structure on the very bottom of this slide, that is a polypeptide chain. And you can see those three amino acids. They're along this whole polypeptide chain. Okay, so in a protein can consist of hundreds, if not thousands, of individual amino acids. This is its primary structure. Just the amino acids in sequence. That is the primary structure. In order for a protein to be a full functioning protein, like what we've talked about in lab, referring back to catalase and how it needed to be in its three-dimensional structure. This is how proteins get into that three-dimensional structure. So first of all, a protein begins as its primary structure. That is its sequence of amino acids. Here at the top of our slide, we've got our amino acid sequence, the three that we were just looking at, how they were put together. Okay, so if we look at how this protein is going to end up becoming functional, proteins end up binding and becoming three-dimensionally shaped because of bonding. Okay, so from the primary structure, that amino acid sequence, these amino acids will begin to fold onto itself, and because of bonding, some of these amino acids can appear to have a corkscrew-like shape, which we call an alpha helix, or they can bend into an accordion-like shape, which is called a beta-pleated sheet, or they can have a random loopy coil appearance. But all of these types of shapes that are form because of bonding between this polypeptide primary structure. Okay, now as soon as the secondary structure begins to take on some three-dimensional shape, then the rest of it begins to pull together, and it's a larger scale structure and more bonding that makes up this tertiary structure, where it's a folded polypeptide structure. Okay, here it's got true three-dimensional shape. Now it's a true protein. Here it's got functionality and it can do something. Now every protein that is functional, regardless of whether or not it's an enzyme or just a simple transport protein or a protein that, like insulin or something that has some sort of function in the body, these proteins, they have to have at least a tertiary shape. But some proteins are really, really big. So some proteins need more than one tertiary structure. So something, for example, like hemoglobin. Hemoglobin is the protein that is in your red blood cells that can bind to 4 different oxygen molecules. It has 4 different tertiary structures. Each one of those can bind to an oxygen molecule. If it didn't have those 4 different tertiary structures, it could only bind to one oxygen at a time. It's a big globular structure. It's called hemoglobin. Okay, so this has a quaternary structure just because it's a really big protein. It's made up of 4 tertiary type structures. Okay, so there are quite a few proteins that are big like that, but not all of them. But every protein that is at least a functional protein has to have a tertiary structure. At a secondary structure, this is not a protein. It's a polypeptide. At a primary structure, it is not a protein. It's a polypeptide. Since we're talking about proteins, and we just finished talking about lipids and carbohydrates, we should talk a little bit about what are called lipoproteins and glycoproteins. So you now know what proteins are. Proteins are amino acids that come together to make polypeptides that then are functional proteins. Lipids are units that are hydrophobic. Glycos are carbohydrates. Okay, so if we look at lipoproteins, lipoproteins are biological molecules that are combinations of those lipids, the lipids that are hydrophobic, and proteins. So these are molecules that are blobs of protein that are surrounded by fat. Okay, so if you can imagine a ball, that's protein, that is surrounded by a covering of fat, sort of like a peanut M&M. Okay, now if you can take that peanut M&M and think of this and refer to that when we talk about HDLs and LDLs. HDL stands for high density lipoproteins, and LDL stands for low density lipoproteins. Okay, these are the lipoproteins that transport cholesterol in human beings. These are important in determining heart disease. So the good lipoproteins are the HDLs. And so what this means is that there is a small amount of lipid to protein ratio. So if you go back to your cross section of your peanut M&M, okay, so with the peanut M&M, the protein is the peanut and the chocolate is the fat. You'll probably never eat another peanut M&M again. Okay, so if you think about an HDL, there's a big peanut inside and a very thin coat of chocolate. Okay, those are the good ones. You want a lot of protein, little bit of fat. The LDLs are a large amount of lipid and a little bit of protein. So you got a tiny, tiny, tiny little baby peanut inside of there and a really big, thick, chocolatey coat. Okay, so there's a bit of protein and a lot of lipid. Those are the ones that are bad for you. Okay, so when you're looking at your cholesterol numbers, the low density lipoproteins, those are the ones that are bad for you. Okay, glycoproteins are a little bit different. These are combinations of carbohydrates. So those sugars, remember those ring molecules, sugars form ring structures, and proteins. Okay, so these tend to form receptors that hang outside of cells. So these are ways that cells are able to talk to one another. And when we start to look at the cell and we look at the plasma membrane, you'll see receptors, and we'll talk about the functionality of those glycoproteins and what those do for each and every cell. Our last topic about macromolecules is about nucleic acids. There are two types of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Okay, DNA is a double strand. Okay, so what you see here is a double strand of DNA. DNA is a polymer. Okay, nucleic acids are polymers, which means that they are made up of individual monomers. The monomers of nucleic acids are called nucleotides. And what you see up here, where the red arrow is, okay, so this is indicating the hydrogen bonds that are occurring between the double strands. Okay, this is the hydrogen bond that is between two nucleotides. Okay, so DNA consists of two strands of nucleotides that are bound together or linked together by hydrogen bonds. So on the left side there is one nucleotide, and then on the right side is another nucleotide. However, the one on the right looks like it's upside down when compared to the one on the left. Okay, each nucleotide is made up of a phosphate group, the P, that is a yellow circle, the red, which is a sugar, and in DNA it's deoxyribose, and then a nitrogenous base, which right here by where the red point of the arrow is, there's a G, that is the nitrogenous base guanine. It is a double ring structure. Okay, so the way that the double helix is put together, the way that it looks on the outside is the sugar phosphate backbone. It's repeating units of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down the outside. So we tend to depict that with a ribbon. Okay, so that's the rails of the double helix. Okay, that's just a repeating units of sugar and phosphate. In the middle, where the rungs of the ladder would be, that is where the nitrogenous bases are. And holding those nitrogenous bases together is hydrogen bonding. Okay, and we'll get to looking at what the nitrogenous bases are here in a little bit. So DNA is housed within the nucleus of eukaryotic cells, which we'll get to talking about cells in the next chapter. It's held in your nuclei as well as another organelle in your cells. But the DNA, which was just covered in the previous slide, is the warehouse for all of your genetic information. It is the blueprint in which those nitrogenous bases encode for all of the information to produce all of the proteins by living organisms. The whole job of your DNA is to produce the proteins that will make you. And of your entire genome, all of your DNA, only 2% of your DNA codes for protein. The other nucleic acid that is similar to DNA is RNA. It is called ribonucleic acid. There are a few differences between DNA and RNA, and one big difference is its sugar is not deoxyribose, but its sugar is ribose, and also its function. DNA is the blueprint that is double-stranded and it's inside of your nuclei. RNA is single-stranded and it leaves the nucleus and it transports the information from your DNA to ribosomes out in the cell, which then make or synthesize proteins. Ribosomes, which make proteins or synthesize proteins, are also made up of RNA. So here's how the process works. Inside of your cells, so imagine this salmon-looking colored square is the perimeter of the cell. Okay, inside of the cell is the cytoplasm, this area where all of the organelles are, and the liquidy gooey stuff in there is the cytosol. Here is the nucleus in which your double strand of DNA is housed. Okay, remember there are two types of nucleic acid, DNA and RNA. DNA is a double strand in your nuclei, and it does not leave unless under special circumstances. Okay, a copy of your DNA is made. That is a single strand, which is this red ribbon, of mRNA. And that RNA is called mRNA because it will take the message of your DNA and it will take it outside of the nucleus. So the mRNA is going to leave the nucleus. Here we see that our mRNA is squeezing through this hole in the nuclear envelope. This is what's called the nuclear pore. So that single strand of mRNA is going out into the nucleus, or out into the cytoplasm from the nucleus. Okay, it's going out into the cytoplasm and it's in search of a ribosome. So the kidney bean shaped things here, there are two of them that come together to make a ribosome. A ribosome is made up of a large and a small subunit. It looks like two kidney beans that come together. Okay, so the ribosome will sort of sandwich the mRNA, and it will read the mRNA, and it will bring in individual amino acids. And if you look down at the bottom of the screen, those individual purple dots, those are amino acids that are being brought in to the ribosome as it reads the mRNA, and a polypeptide is being synthesized, being produced. That polypeptide is the primary structure. As that primary structure is being built, it begins to bend and twist and fold on itself. It will then become its secondary structure and then eventually become a tertiary structure. Hence, the production of a protein. Voila!